Growth factors for animal cell culture. In today's lecture, we'll discuss about the growth factors or growth hormones which are essential for the successful establishment of animal cell culture. A growth factor is a naturally occurring substance capable of stimulating cellular growth, proliferation, healing, and cellular differentiation. Usually it is a protein or steroid hormone. Growth factors are important for regulating a variety of cellular processes. Let's discuss the important growth factors in detail. EGF. Epidermal growth factor, EGF, stimulates cell growth and differentiation by binding to its receptor, EGFR. Human EGF is a 6-CADA protein with 53 amino acid residues and 3 intramolecular disulfide bonds. Initially, human EGF was known as urogastrone. EGF was originally described as a secreted peptide found in the submaxillary glands of mice and in human urine. EGF has since been found in many human tissues including submandibular gland, submaxillary gland, and parotid gland. The production of epidermal growth factor has been found to be stimulated by testosterone. Functions of EGF. EGF, binding to EGFR, results in cellular proliferation, differentiation, and survival. Salivary EGF, which seems to be regulated by dietary inorganic iodine, also plays an important physiological role in the maintenance of oroesophageal and gastric tissue integrity. FGF. The fibroblast growth factors are a family of cell signaling proteins that are involved in a wide variety of processes, most notably as crucial elements for normal development. Any irregularities in their function lead to a range of developmental defects. These growth factors generally act as systemic or locally circulating molecules of extracellular origin that activate cell surface receptors. A defining property of FGFs is that they bind to heparin and heparan sulfate. In humans, 22 members of the FGF family have been identified, all of which are structurally related signaling molecules. Functions of FGF. FGFs are multifunctional proteins with a wide variety of effects. They are most commonly mitogens but also have regulatory, morphological, and endocrine effects. They have been alternately referred to as pluripotent growth factors and as promiscuous growth factors due to their multiple actions on multiple cell types. PDGF. Platelet-derived growth factor, PDGF, is one of numerous growth factors that regulate cell growth and division. In particular, PDGF plays a significant role in blood vessel formation. Platelet-derived growth factor is a dimeric glycoprotein that can be composed of 2 subunits, PDGFR, to B subunits, PDGFBB, or one of each, PDGFAB. PDGF was one of the first growth phases characterized and has led to an understanding of the mechanism of many growth factor signaling pathway. The receptor for PDGF, PDGFR, is classified as a receptor tyrosine kinase, RTK, a type of cell surface receptor. 2 types of PDGFRs have been identified, alpha type and beta type PDGFRs. Functions of PDGF. PDGFs are mitogenic during early developmental stages, driving the proliferation of undifferentiated mesenchyme and some progenitor populations. PDGF plays a role in embryonic development, cell proliferation, cell migration, and angiogenesis. Overexpression of PDGF has been linked to several diseases such as atherosclerosis. PDGF is a required element in cellular division for fibroblasts. PDGF is also known to maintain proliferation of oligodendrocyte progenitor cells. NGF. Nerve growth factor, NGF, is a neurotrophic factor and neuropeptide primarily involved in the regulation of growth, maintenance, proliferation, and survival of certain target neurons. NGF is initially in a 7s130 KDA complex of three proteins alpha-NGF, beta-NGF, and gamma-NGF in 2 hours 1 minute and 2 seconds ratio when expressed. This form of NGF is also referred to as pro-NGF, NGF precursor. NGF binds with at least two classes of receptors, the tropomyosin receptor kinase A and low-affinity NGF receptor. Both are associated with neurodegenerative disorders. Rita Levi-Montalcini and Stanley Cohen discovered NGF. Functions of NGF are neuronal proliferation, proliferation of pancreatic beta cells. NGF plays a critical role in the regulation of both innate and acquired immunity. In the process of inflammation, NGF is released in high concentrations by mast cells. Ovulation, NGF is abundant in seminal plasma. Recent studies have found that it induces ovulation in some mammals. Erythropoietin. Erythropoietin, also known as hemopoietin or hemopoietin, is a glycoprotein cytokine secreted by the kidney in response to cellular hypoxia. It stimulates red blood cell production, erythropoiesis, in the bone marrow. Erythropoietin is produced by interstitial fibroblasts in the kidney in close association with peritubular capillary and proximal convoluted tubule. It is also produced in perisinusoidal cells in the liver. Liver production predominates in the fetal and perinatal period, renal production predominates in adulthood. Exogenous erythropoietin, recombinant human erythropoietin, RHEPO, is produced by recombinant DNA technology in cell culture and are collectively called erythropoiesis-stimulating agents. Two examples are apoietin alpha and apoietin beta. ESAs are used in the treatment of anemia in chronic kidney disease. Functions of erythropoietin. Red blood cell production. Erythropoietin is an essential hormone for red blood cell production. Interleukin-1 or simply called as IL-1. Interleukin-1 alpha and interleukin-1 beta are cytokines that participate in the regulation of immune responses, inflammatory reactions, and hematopoiesis. Interleukin-2 or IL-2. These factors, which include interleukin-2, are secreted by lectin or antigen-stimulated T cells and have various physiological effects. IL-2 is a lymphokine that induces the proliferation of responsive T cells. In addition, it acts on some B cells via receptor-specific binding as a growth factor and antibody production stimulant.
Thanks for watching and subscribe to our channel, which made science easy to understand.